I'm Dr. Sophie Holland, and this is my lecture about a comprehensive method for analysis of polystyrene microplastic biodegradation in seawater. So, we are looking at microplastics in marine environments, which is a huge problem globally, as we all know that there is a lot of plastic contamination in the ocean, and as well as primary microplastics, bigger pieces of plastic continue to break down new microplastics all the time. So we've got a big issue with marine pollution on a global scale. Polystyrene is one of the key polymers that is found in the ocean, and frustratingly, it's also one of the most resistant to biodegradation. There are limited studies on biodegradation of polystyrene, particularly within the marine environment, and one of the main hindrances of these studies is a suitable method to analyze whether polystyrene is being degraded, aside from just a sort of relatively simplistic weighing in before and afterwards, which is obviously also incredibly difficult when you're working with microplastics as opposed to larger pieces of polystyrene. The overarching aim of this work, or the end goal, is to be able to do DNA stable isotope probing with a 13C labeled polystyrene, but in order to do that in microcosms and know when it's time to harvest the DNA to do the SIP, we need to know when it's being degraded and when is the optimal time to take those DNA extractions. So, to develop a straightforward and reliable method to investigate polystyrene degradation, we decided to try and look for degradation products within some seawater microcosms. We took seawater from near the Heriot-Watt Channel in the northeast Atlantic, which is an area that has a lot of oil and gas production, kind of hoping here that we would see maybe plastic degraders because there's likely to be maybe a slightly increase in hydrocarbon degraders given the abundance of oil and gas in the area. Seawater was taken from a depth of 150 meters, brought back to the lab, filtered through a 120 micron filter and put into two 50 ml serum bottles along with some marine supplements which just contain trace elements and vitamins, as well as our polystyrene. So we had 6 treatments that had unlabeled, just regular 12C polystyrene, and 6 that had our 13C labeled polystyrene. Every week, liquid samples were taken of 1 ml, and we also exchanged the headspace with air in order to keep it aerobic. From the liquid samples, we then did a liquid-liquid extraction to get a total lipid extract, and that was to remove debris like salts and also aqueous media so that we could run these nicely on a GCMS. So we had a liquid sample, and we turned it into a total lipid extract using the method of Bly and Dwyer, or a DCM methanol extraction. When we initially ran the total lipid extract on our GCMS, we didn't really see a lot of peaks, so we then further drilled down into that, and we did a acylation derivatization PSTFA. So we got trimethylsilyl derivatives, which should target the kind of smaller aromatic compounds that we thought we were interested in. Now, the 4 compounds that we chose here, 2 of them are based on previous reports in the literature, which is 1-phenyl, 1,2-ethanediol, and 2-phenethanol. The other 2 are just based on kind of looking at the polystyrene molecule, seeing a bunch of benzene rings attached to the carbon backbone, and presuming that if we're going to get microbial degradation, you're probably going to see stuff like phenol and catechol coming out. So we ran samples from the 12C or unlabeled polystyrene on our GCMS. We got this nice total ion chromatogram. We then drilled down into that a little bit to look for the ion sizes that we kind of expected to see based on our deduced products, and took extracted ion chromatograms. We also looked at the mass spectra and found ions of interest. One way that we helped to identify peaks of interest was to look for 73 mass ions, because that is the weight of the mass of the trimethylsilyl piece, which breaks off quite easily after derivatization. That gave us peaks of interest to look at as well. So basically we did see all 4 of these compounds, but to varying degrees of certainty. So for example, the 1-phenyl, 1,2-ethanediol does come out as like a peak that we can see on the TIC, and a very clear peak on the XIC. We also see all 3 ions of interest at relatively high relative intensity in our mass spectra. Other peaks are less certain, for example, the 2-phenylethanol, which is a very, very small peak in our TIC and barely makes a blip on the zoomed out XIC, but is there when you zoom in, I promise. When we apply this analysis to the time series that we have thus far for the 12C samples, interestingly, we did see all compounds present at time 0 and then tending to decrease, which is not what we expected. However, the method is still slightly under development. We're still corroborating with our controls, for example, the no polystyrene control, and we're also in the process of getting standards so that we can be sure that these compounds we're seeing are what we think they are. So, benefits of this method, are that it is highly sensitive. So we started with 10 micrograms of polystyrene in a 100 ml seawater, and given the rate of polystyrene degradation is expected to be very, very low, and this data was up to 42 days, we can expect that we're really looking at like sub ppm levels of these degradation products here. So the fact we're seeing anything at all is pretty cool. And when we get this kind of completely verified and working, we will then be able to do the same thing with our 13C samples. What's exciting about that is that the GCMS will allow us to identify what the compound is, and then using the IRMS, we'll be able to show that there is incorporation of 13C by the delta 13C enrichment factor. So we'll be able to show that yes, this is a compound that we're seeing, and yes, it has come from polystyrene. The other cool thing about having a total lipid extract is we can do a different derivatization. We can do an acid methanolysis to get a fatty acid methyl ester and look at phospholipids in the cell membrane, and then show incorporation of 13C from polystyrene into the bacterial cell membrane. And on top of that as well, we do have the SIP, which is ongoing, so we'll hopefully be able to show as well that based on DNA stable isotope probing, these bacteria are responsible for breaking down polystyrene. This is what they're breaking it down into, and they are incorporating it into the cell membrane. We think this is a very useful method, especially given the kind of absence of straightforward GCMS methods in the literature at the moment for detecting biodegradation or abiotic degradation of polystyrene. Thank you to EVNet for funding the travel bursary to get me here.